A very good afternoon to everyone. Welcome in the class. And as we see in the starting, let's crack CSIR UGC NET. And that is the name of our channel. Let's crack CSIR UGC NET. Before beginning with the topic, let us have a small introduction about Unacademy and about myself. Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET. I'm your educator Farida Bandookwala, and I have a teaching experience of 10 years and I have a teaching experience of 10 years and I've qualified MP-SLET examination in year 2018. If you're opting for Unacademy, what all things you are going to get? You're going to get daily life classes. The whole syllabus is quite structured so that you can complete the whole thing in a defined span of time. Then live tests and quizzes are taken so that you can evaluate that how much you have learned so far, and unlimited access towards all the videos which are recorded in the category. Then free live classes are conducted on two platforms. One is YouTube. For taking the YouTube class, you just need to follow the account and click the bell icon for getting all the notifications. And another kind of classes are special classes. For special classes, what you need to do is you need to download the Unacademy Learning app. When you will download the Unacademy Learning app, then you will be asked to fill the referral code of any educator. So fill my referral code, that is FP01, and then you will be directed towards the special classes. You can watch my all the previous classes and you can watch all the upcoming classes. Recently a discount is going on in the plus subscription, and this plus subscription is a paid subscription, and all the topics of CSIR are there in this plus subscription. So by just a second. So by opting for plus subscription, you will be getting all the videos in a well systematic manner on the platform of Unacademy. So recently a discount is going on, and for 12 months you need to pay 11,550 rupees, and for 24 months you need to pay 16,800 rupees. You can get an additional 10% discount if you're using my referral code, that is FP01. So do not forget to use my referral code FP01. For 12 months it is going to cost you 10,395, and for 24 months it is going to cost you 15,120 rupees. So that was a little about Unacademy. Let us talk about the topic of today, that is gene isolation. Gene isolation. This is the topic of today. So there are basically three main methods for obtaining genes. So the three methods we'll be discussing in detail. First method is synthesizing the gene using an automated gene machine. Synthesizing a gene using automated gene machine. Then second is gene cloning. Third is with the help of PCR machine. These three are the methods of gene isolation. So first of all, we'll be talking about gene machine. So gene machine is a fully automated commercial instrument. It's this gene machine, the word itself is quite clear that some machine is used. So it's a fully automated machine. Fully automated machine, and it is also called as automated polynucleotide synthesizer, also called as automated because it is completely automatic. So automated polynucleotide synthesizer. And the word itself, gene machine. So these are its synonyms, and it is available in market which synthesize the predetermined polynucleotide sequence whichever we want to. So which can synthesize a predetermined polynucleotide sequence. The genes can be synthesized rapidly and in high amount with the help of gene machine. Genes can be synthesized rapidly and in high amount with the help of gene machine. Now, for example, that we have a gene sequence for tRNA. So it can be synthesized in the gene machine in few days. So it's a simple method, almost correct method, and it is also a rapid method. It synthesizes short segments of single stranded DNA under the control of microprocessor, so which can be used as primers and probes. Can automatically synthesize short short segments of single stranded DNA. So the working principle of gene machine, we'll see that. So how it happens that development of insoluble silica-based support in the form of beads, which provide the support for solid-based synthesis of DNA chain that is needed. The first thing that is needed is insoluble silica-based support. Insoluble silica-based support. For giving a base for the synthesis of DNA that is needed. Then development of stable deoxyribonucleotide phosphoramides as synthons which are stable to oxidation and hydrolysis and ideal for DNA synthesis. So second one is important, development of stable deoxyribonucleoside deoxyribonucleoside phosphoramides as synthons which are stable to oxidation and hydrolysis and ideal for DNA synthesis. Now, four separate reservoirs containing the nucleotides, that is A, T, C and G, they are connected with a tube to a cylinder packed with small silica gels. I'm going to draw a description picture as well. First, you just listen to theory. So, what is that? Four separate reservoirs are needed. Four separate reservoirs. Four separate reservoirs of A, T, C and G are connected to a tube to a cylinder which is packed with silica beads. Connected with tube to a cylinder packed with small silica beads. So we'll just draw this what I told you. See, there will be microprocessor control in the very starting. Microprocessor control. And then it will be connected with all the other things like this. Like this. And these all things, these are the wires which are giving the instructions. Like this. Like this it will be connected. So here we can write GCTA. These are the nucleotide reservoirs. Nucleotide reservoirs. And these are the reagents and solvents needed. Reagents and solvents needed. Right. Now they are going to go into the cylindrical tube which I told you, which is over here. There will be a pump over here, pump over here, and they are going to move in this direction. Okay, and in this tube there will be silica beads. These are the silica beads. And this column is also called as synthesizer column. Right, and this is the collector tube, collector, and this is for waste. So this is how the whole processing of gene machine looks like. Right, so we have seen till 4 steps. Third steps, that 4 of them are in the separate reservoirs. After that, let us talk about the beads. Beads, they provide support for the assembly of DNA molecules. So reservoirs and reservoirs for reagents and solvents are also attached along with it, right? So reservoir for reagents and solvents are also attached. All the reagents and everything that will also be given. Now the whole procedure of adding and removing the chemicals from the reagent reservoir in time is actually controlled by microcomputer control system that is called as microprocessor. So what is the function of microprocessor? Controls addition of reagents and nucleotides in the tube for synthesis. That is the function of microprocessor. After that, if one desires to synthesize only polynucleotide with a sequence of nucleotide T, G, C and without any A, the cylinder is first filled with beads with a single T attached. So that is how the thing are going to go. and after that it will be flooded with G, and the remaining of the G's will be then flooded with C. So that will be the sequence. So if one desires a specific sequence with a specific thing, they have to go this way. So if one desires to synthesize a short polynucleotide with sequences of T, G and C, the cylinders will be first filled with the beads with a single T attached. After that, they'll be flooded from the reservoir. The right hand side of each of the G is blocked by using chemicals from the reservoir so that its attachment with any other G's can be prevented, and the remaining G's could not join with the T and are flushed from the cylinder. So the other chemicals are then passed from the reagent and the solvent reservoir so that these blocks can now be removed from the G, which is attached to the T. And in the next cycle, C will be flooded and C would be blocked again so that no two C's can be joined together. And when C will join with T, G and C sequence, then there will be flooding of the reagents, which will be actually responsible for removal of all kind of blocks and joining of further sequences. That is how the whole thing moves. So I would write over here cycles of overflooding and removal of blocks will be performed. Removal of blocks would be performed. This is how the things occur. Now the desired sequence is entered on a keyboard and the microprocessor automatically opens the value of nucleotide reservoir, chemical and solvent. Whichever sequence we have given, that will be responsible for opening and closing of that particular reservoir. So in gene machine, the nucleotides are added into polynucleotide chain at a rate of 2 nucleotides per hour, not more than that. So rate is very important. In gene machine, we'll take another color. In gene machine, the nucleotides are added. The nucleotides are added into polynucleotide chain at rate of 2 nucleotide per hour. So by feeding the instructions of human insulin gene in the gene machine, human insulin can be synthesized for whichever nucleotide sequence we want the gene. It can be easily synthesized in the gene machine. So this was the first method of gene isolation, that is to synthesize the gene. It can also be called as gene synthesis. After that comes as gene cloning. So gene cloning is a common practice in molecular biology labs that is used by researchers to create copies of particular genes for downstream applications such as sequencing, mutagenesis, genotyping, or heterologous gene expression or protein synthesis, etc. These are some of the applications of gene isolation through the help of gene cloning. So the traditional technique for gene cloning involves transfer of a DNA fragment of interest from one organism to a self-replicating genetic element such as bacterial plasmid, and this technique is commonly used today for isolating long or unstudied genes and protein expressions. So we'll be seeing the sequence for this. First of all, we'll be talking about the introduction which we have discussed just now. So it's a common practice in molecular biology labs and it is used by researchers to create copies. Creates copies of a particular gene, for sequencing, then mutagenesis, then genotyping, and heterologous expression of protein, etc. So these are some of the things why gene cloning is performed. And the traditional and unique technique for gene cloning involves transfer of a DNA fragment from the interest organism into the self-replicating genetic organism such as bacterial plasmid. And these techniques are widely used for isolating long and unstudied genes. So used for isolating long or unstudied genes and protein expressions. This is the thing. After that, we'll be seeing the sequence, that how it is performed, that is the steps of gene cloning. Steps. So gene cloning is a very popular technique. We'll be doing a separate video also on this. We'll be seeing this in a very short flow diagram way. This is a vector, then this is a fragment of DNA. So I will draw the fragment of DNA in different color. This is the fragment of DNA. Fragment of DNA. This is how recombinant DNA molecule is obtained. rDNA molecule. After obtaining rDNA molecule, it will be introduced into the bacteria where it can be multiplied. After introduction of this into vector, there will be transport into the host cell. So second is transfer of this whole thing into the host cell. So this is how it is transferred. This is how it is transferred. So this is the transport into host cell. Transport into host cell. Now after this, multiplication of recombinant DNA molecule. So multiplication of rDNA molecule. This is various copies of the rDNA which we prepared. So multiplication of rDNA molecule and then division of host cell. Division of host cell. Division of host cell with lots of copies of rDNA, and after that numerous cell division resulting into clones. It will give rise to clones, and this is how the gene isolation can be performed easily with the help of cloning technique. So we know the restriction enzymes which will be cutting this rDNA obtained from the bacteria, so it can be easily obtained that way. That is, now we have prepared the multiple copies, and the question of demand of multiple copies is actually solved. After that, the next method for gene isolation is PCR. We have done a separate video on PCR in detail also, but we'll be here talking it in terms of the gene isolation, in light of gene isolation, though we have done a detailed PCR on the same channel, so you can anytime watch that. The polymerase chain reaction can be used to obtain pure sample of gene. Used to obtain pure sample of gene. Pure sample of a gene. After that, it is because the region of starting DNA molecule that is copied into the PCR segment, the boundaries of it is known. So it can be very much pure. So because of the sequences are known, and there are primers which are used for this, they give rise to a pure sample. Now, if primers they anneal at the either size of the gene of interest, many copies of that gene can be synthesized. So outcome is the same as we do with the gene cloning experiment. Same thing will be obtained. Although the problem of selection does not arise because desired gene is automatically selected. We don't have to select for it. It is automatically selected, and as a result of that, in the end, we are just going to get the desired gene. So the end product is the end product is desired gene only. Why? See, if this is the whole DNA and we are providing primers only for a particular sequence, only for this sequence we are providing the primers. So what it will do? It will do the extension of only this much in the first cycle. In the second cycle, it will be something like this. Oh, sorry. It will be hybrid like this. So in second cycle, again we are going to get in the second cycle, we are going to get many copies of the newly synthesized gene sequence. Like this we are going to get, and there will be very less hybrids. But still, if hybrids are also there, then too they are just giving rise to the desired gene only. We are going to get desired gene in the end. So an alternative to cloning, polymerase chain reaction can be used to directly amplify the rare specific DNA sequences in a complex mixture where the end sequences are known. So it's a very much easier, convenient and foolproof method to perform the PCR. Now there are certain limitation, as we are saying that PCR is very important, very interesting, and pure samples can be obtained and all. We are like praising PCR since it's starting because it is quite easy also. We just need to take the whole DNA, just DNA isolation is needed and primers are needed, and rest all of the things will be performed in the machine. So we are just praising PCR so much, so we need to know that is there any limitation also associated with PCR. So we will be talking about limitations of PCR in light of gene isolation. So in order for the primers to anneal to the correct position, to join at the correct position, either side of the gene of interest, the sequences of these annealing site must be known. Without knowing, it cannot be performed. So it is easy to synthesize a primer with predetermined sequence, but if sequence of the annealing sites are unknown, then the appropriate primers cannot be formed. So this means that PCR cannot be used to isolate the genes that have not been studied before, and that has to be done. So you can write in limitation, cannot be used for genes whose whose sequences are not known before. So only the sequences whose only for the genes whose sequences are known can be used for this first limitation. Then there is a limit to the length of the DNA sequence that can be copied by PCR. 5 kilobases can be copied very easily, and segments up to 40 kb can be dealt with using specific techniques. But shorter the length of the genes, specifically for humans and vertebrates, cloning must be done for the smaller sequences. So we can write another limitation: cannot copy bulky sequences. Cannot copy bulky sequences. Short sequences, 5 kilobase pairs to 40 kilobase pairs can be done, but above 40, more than 40 kb, results are not accurate, are not accurate. So this is how we have seen the gene isolation in three methods. That is, first of all, through gene machine, second one was gene cloning, and third was PCR. So gene machine, we have read that it is a fully automated machine. It is also called as automated polysynthesizers. In this, the gene can be made in a rapid manner in high amount, and automatically this gene will be synthesized. Just we need to provide the nucleotides. So this is how the machine or the processing looks like. This is the microprocessor control, or you can also write computer, from where the whole instruction of the sequences would be given, that which nucleotide has to be added next. Wherever the sequence or wherever the signal reaches, that reservoir is going to open, and that type of nucleotide is going to flow into the cylindrical tube. The cylindrical tube contains lots of silica beads, and on silica beads the newly synthesized sequence would be attached. So wherever the signal reaches, there it opens and it attaches over here. And every time whenever it opens, these nucleotides, they have got a block in their right. Left will go and attach the bead, but right would be blocked. It cannot attach with one more sequence of G, because there will be lots of chaos. So in that case, they have provided a block, and blocks can be removed by these reagents and solvents. So after addition of a nucleotide, some unblocking agent would be provided. When that unblocking agent is going to unblock the right end of this nucleotide, then only that nucleotide will be able to join with the other nucleotide. That is the thing in the processing of gene machine. And this is the tube where the nucleotides, they flow and they attached on the silica beads in this cylindrical tube, right? So these are the things where the reagents and solvents are stored, and these are the places where nucleotides are stored. So these that was the principle. And in gene cloning is used for producing the sequencing and mutagenesis and genotyping. So for gene cloning also, we have seen the steps that vector is taken, fragment of DNA is taken, and they both are joined, and rDNA molecule is made, and then it is introduced in bacterium. It is led for multiplication, and then many copies of the bacterium are made. So n number of copies of the genes are obtained. And then we have seen that PCR, that only specific sequence would be amplified. So it is very specific, and only desired gene is obtained. We don't have to do much of the thing, just we need to isolate the DNA. But the limitation is that only the gene sequences whose sequences are known to us, only those sequences we can amplify. And another limitation is it cannot copy bulky sequences. It can copy only small segments and sequences. So like this, we have completed gene isolation by 3 methods, and I hope you people have understood the thing well. And as we say in the end, that let's crack CSIR UGC NET and GATE 2021 together, as a crash course is going on for GATE 2021. So let's crack these examination, and hit the bell icon for getting all the notifications of the upcoming videos. If you really like my video, give it a thumbs up. It really motivates the educators like us. And for getting an additional 10% discount in the class subscription, do not forget to use my referral code, that is FP01, and you can use my referral code for attending the special classes as well. For special classes also you will need the referral code. So by using my referral code, you can attend my classes as well as you can attend the classes of other educators as well. And it was great teaching to you all. Thank you for your patient listening. I hope to see you all tomorrow at 10:30 a.m. in the morning and 5 p.m. tomorrow in the evening. In the morning we'll be doing a topic of CSIR syllabus, and in the evening we'll be doing a topic of GATE syllabus. So this is it for now. Thank you very